in 2 hours' time here. Basically this will be a short talk with not too much data because I've just started in the Egan lab a month ago. However, I have been interested in the use of eggs as bags of reprogramming factors for quite a long time. I earned my PhD in the Roslin Institute working with Jim McQueer and Ian Wilmot, and they were they were the team that cloned Dolly the sheep. So, and subsequently, I was interested that led me to aspects of epigenetic reprogramming, and I traveled to Cambridge where I worked in Azim Sarani's lab looking at epigenetic changes occurring when you restore developmental potential to somatic cells. So this first slide is just an introduction to reprogramming. Reprogramming is the process by which developmental potential is restored to a differentiated somatic cell. And in the Egan lab at the moment, what we're trying to do is trying to find the epigenetic changes that are concomitant with the reprogramming that you see when you fuse somatic cells to human ES cells. Because the somatic chromatin is reprogrammed back to a state very, very similar to the human ES cell state. So I think when we learn more, eventually we could see a time where we would be able to, we would have identified the mechanisms underpinning nuclear reprogramming, and we would be able to manipulate them. And this is just the dream slide. When we have this information and we can manipulate the system, we could see a time where we have a patient with a disease, and we could take some easily a small number of cells that are easily biopsyable from the patient, and take those and bring them to the lab, manipulate the epigenetic mechanism so that nuclear reprogramming takes place, and that you get reprogramming of your somatic cells back to your pluripotent stem cells. And apart from pluripotent stem cells being able to give you a range of different cell types, they're also very useful in that you can expand them in culture. So obviously if we had to take large amounts of cells from a sick patient, that wouldn't be, I suppose, a plausible therapy. But we can, just by taking a small number of somatic cells and reprogramming them back to this pluripotent stem cell-like state, we can expand them, and then if we had the necessary information, we could direct differentiation into a specific cell type that the patient requires. And then we could introduce those back into the patient, at least in theory, and because they're originating from the patient, you'd avoid various immunorejection problems. So this process of nuclear reprogramming has been studied in several different ways. I think the really most vigorous way of showing restoration of developmental potential is to take an adult somatic nucleus and put it into an enucleated egg or ooplast. And then you can show by the production of a viable animal that you have indeed restored developmental potential. But nuclear transfer is quite laborious. It's a very difficult technique to learn, and the main problem, I think, is a question of whether you can generate enough material to really go after the genetic changes that are responsible for reprogramming. So I've been using a different system whereby I'm fusing embryonic stem cells to adult somatic cells, and then selecting for reprogramming, selecting for reprogrammed fusion events. So this is just the system that I've been using for many years. The diagrams are all of mouse cells or mice. We've moved on now, we're doing this in human cells. So just as a bit of introduction of jargon, when you fuse one cell type with a different cell type, you get what's known as a heterokaryon. And this is basically two nuclei sharing the same cytoplasmic bag, if you will. And what you can get, in rare instances, you can get nuclear fusion forming a tetraploid hybrid cell, assuming that the two fused cells are diploid. So when you fuse a mouse ES cell and a mouse somatic cell, and then apply antibiotic selection, because fusion is quite low between your two cell types. This is one of the key problems that we're addressing at the moment. The two problems are we need to increase cell fusion, and we also need to increase the incidence of reprogramming. We really want to, all these processes are quite inefficient, and in order to study this, we need more material. So traditionally what people have done is they've fused somatic cells, ES cells, and then put them under antibiotic selection so that you can select for fusion products. So you get something like this, which is an ES phenotypic hybrid colony. Now, the problem, I suppose, with this approach is that it takes several weeks for these hybrid colonies to grow, and you have enough material to analyze. So people have looked at the epigenetic changes in these cells, but they're looking at epigenetic changes that could well have occurred after reprogramming. So they're not directly important for the induction and perpetuation of reprogramming to an ES stem cell state. So in order to try and address this problem, what we've done is we've using OCT4 GFP somatic cells, and so in these somatic cells, OCT4 is a pluripotency gene which is off in differentiated cell types and on in pluripotent stem cells and germ cells. So if we take these cells, they'll be GFP negative because this gene will be off, and then we fuse it to ES cells, and we can isolate, we can identify reprogramming as green fluorescence appearing in our fusion products. So this is the way we're now attempting to get to reprogramming much sooner, much sooner than previous studies. So this is just some preliminary data where we're showing that reprogramming, the first evidence of reprogramming is occurring as soon as 18 hours after fusion. Now you can actually see the protein GFP fluorescing here in small numbers. So when you fuse an ES cell to a somatic cell, reprogramming actually happens surprisingly quickly. So this slide refers back to the second problem to which I alluded to. If we're to get enough material, we're going to have to increase the incidence of reprogramming. This just shows you can actually treat the cells with drugs that will render a more open chromatin structure, and this would make reprogramming more easy. The problem with some of these drugs is that they're quite toxic. So even though you might get reprogramming more frequently, the cells die quickly. So actually the use of these drugs is questionable. One process we're investigating at the moment is we're going to use C mice, which Kevin Eggen has generated in Rudolf Janisch's lab. And these mice are quite interesting because they're viable, but they have their DNA hypomethylated. So DNA methylation is associated with gene silencing and a closed chromatin structure. So we believe that if we take somatic cells from these particular mice, that they will be easier to reprogram. This slide, I suppose, contains 4 histograms, but it represents a good 1.5 years of hard slog during my PhD, because unfortunate for me, human ES cells are far less fusogenic than mouse ES cells, and I spent 1.5 years trying to increase their fusogenicity, and tried many different things. And this is just to show you that if we take down the hyaluronan component of the extracellular matrix of human ES cells, we can increase cell fusion 5-fold. And this is significant because it now gives us enough material to start studying the epigenetic changes that occur during reprogramming. Obviously, because I came from Azim Surani's lab in Cambridge, you could say that I'm studying an artificial process with nuclear transfer or with the cell fusion. I think in order I suppose, in order to show that some of what we learn from these epigenetic changes during reprogramming, that there is something consistent or something similar to it in vivo, we need to study epigenetic reprogramming in vivo. And this is the way we have done it in mice. So we extract primordial germ cells, as they migrate, epigenetic reprogramming occurs. So this is an in vivo system in which we can look at, see if the same things are happening in vivo and in vitro. I put this figure up because I think I've worked in 5 different human ES labs now, and every time you go in and look at the cells always have different characteristics, and people have vastly different culture conditions and ways of treating the cells. And I think one of the first steps we really need to achieve is homogenization of the culture conditions that everybody's using, so that we can increase repeatability of data produced between labs. And finally, I'm just going to allude shortly to a project that's not, well, I suppose it's not directly related to nuclear reprogramming, but obviously if it did succeed, it would be very useful as a tool. So we're trying to generate human germ cells, early human germ cells, from human ES cells. And the idea is to differentiate the ES cells as embryoid bodies, and then dissociate those cells and sort for cells that have early germ cell markers. And then we'd culture those cells and look for later germ cell markers. The idea is that, so we're trying to find out whether you can recapitulate normal development in the dish. So this is just some preliminary data with human ES cells. We've differentiated them for 14 days, differentiated them, and then sorted them here using c-Kit antibody. So it's early germ cell marker. And we would isolate that and then subsequently try and culture it on and get other later markers of germ cell development being switched on. So this is the guy who put me in this situation today, Kevin Eggen. So hopefully I've earned some brownie points, and I think that's it. You can tell that I'm an unexperienced speaker. I forgot to thank Anne and Keith for inviting Kevin and for leaving me in the door.
You're forgiven.
Thank you.
Welcome.
Questions?
I was wondering, have anybody in your lab ever tried doing nuclear transfer with these cells that you grow in to reactivate alpha and nano?
Yeah.
Do you have easier nuclear transfer frequency? Success rate?
With EC cells?
With the cells that you grow so that you can reactivate the alpha and G. You grow in a drug, in presence of drug, so you can reactivate alpha and nano?
Yeah, yes, I mean, that we would try that, but you see the thing is, well, when I did those experiments, the cells really, there's a high incidence of cell death, like 48, 72 hours afterwards. So I think we were going to try that approach, but then when we came up with this alternative use of this chip C mouse, which is hypomethylated, this seemed like a more promising approach, so we switched to that.
Yeah, hi. Are they on MEFs, and are you taking in consideration passage number? Because, you know, there are groups that have been finding there's epigenetic changes just in passaging as well.
Yes, I think ideally you'd like. I mean, when I was in Roslyn, I never grew the cells on feeders. I suppose it depends. I mean, initially we're just going to be answering some small biological questions. We're not maybe too worried whether they're exactly perfect ES cells. So for my preliminary experiments, I'm not going to do it in a few years. But hopefully, in a few years' time, we'll be able to grow human ES cells in purely, defined conditions. So just a few chemicals added, and that will be when.
But again, if you're taking a dish, just even with H9s, you have ones that look undifferentiated, ones that are slightly differentiated. So that would make a big difference which ones you would pick for fusing.
Yes, I mean, you'd obviously have to sort to get a good undifferentiated population, and you can use the FACS sorter to do that. But I think the lines, Doug Melton's lines, the lines that I've seen so far, I mean, they grow. We've got 1 or 2 lines growing as a reasonably good undifferentiated monolayers now. So.
Yes, Stephen. If you fuse a skin cell with an ES cell, okay, we know that the pluripotency genes in the skin cell light up.
Yeah.
We also, I think, know that some of the skin genes in the skin cell remain on. They don't all switch off, at least at the same time. Now, I wondered whether that hybrid cell would differentiate more easily into skin than into, say, liver and neural cells.
Well,
does it remember? Is there a memory here?
Well, that's a good question, and I suppose it's hard to differentiate an active memory mechanism from just incomplete reprogramming.
Well, the incomplete reprogramming would be, as it were, the basis for the memory.
Yeah.
But it would be interesting to know if the memory was there.
So, just your question was like, are different cells, possibly from the different germlines, do they have different amenabilities to being reprogrammed? And indeed, we plan to do fusion experiments. But I mean, again, the other thing we're trying to address is whether you can just reprogram human somatic cells with cytoplasts. So, I mean, in these hybrids, the ES cell-derived chromatin is still present. So maybe you could say that it's factors being produced from the ES cell-derived chromatin are keeping the somatic cell in a reprogrammed state, if you will. So in order to truly show, I think, reprogramming, we need to get rid of the DNA from the ES cell in the hybrid.
What about the reverse question? Are there different reprogramming cells? So could it be that somatic and stem cell reprogramming factors are not the same than those present in a normal cell?
That could be true. I mean, one of the interesting things that I found in my thesis was that the longer you have cells in culture, ES cells, the more they lose reprogramming activity. So, I mean, one approach maybe we could see there is using high passage and low passage ES cells and looking for different genes expressed and see if we can find a correlation.
Maybe you can also use cytoplasms from oocytes.
Yes, yeah.
And use those to compare size relationship between volume and size.
But if you're going to do that in a human, it's a lot easier to go into the lab and grow the cells up as opposed to nagging women for eggs.
I also have a question, hybrid cells. So you look at the activation of from somatic cells. Have you paid any attention to see if somatic cell will
that's
ES cell into that?
The experiment I was doing this morning before I was interrupted. So I'll explain that experiment to you because it's fresh in my mind. So we've got an ES cell that has an albumin promoter, so a liver promoter driving GFP, and we're fusing that to a liver cell, a hepatoma cell actually, that has REX1, which is pluripotent stem cell promoter, driving DS-RED. So we fuse those together. And then presumably we would get 4 populations, one where neither promoter is activated, one where the somatic promoter is activated, one where the ES cell promoter is activated, and one where maybe there's a war going on between the 2 programs. So once I get that information, I'll be able to answer your question.
I wanted to ask a question that goes back to bitterly fought issue about whether the donor cell is or isn't in the cell cycle in terms of its efficacy and being reprogrammed and being in cloning being a donor cell.
Okay.
So your donors, have you looked to see whether if you arrest the cells in G0 whether they, and then fuse them, or whether they have different fates than if they're just randomly cycling cells?
Well, using mouse ES cells and fibroblasts during my thesis, in Rosalind, various people had a patent saying that it was really serum starving the somatic cells into quiescence that was important. But I have to say, when I looked at the cell cycle characteristics, there was no clear relationship between the two. If you actually took serum starved cells and fused them to ES cells, you got a very high level of reprogramming. But when you took those serum starved cells, put them back into full serum, and then look at them, so they've begun cycling again, so they've re-entered the cell cycle, they still have elevated reprogramming. You see in those hybrids, you see, so they're still more easy to be reprogrammed, more easily reprogrammed. So I don't know if there's a direct
They're
correlation.
simply randomly grown.
Well, I would imagine that when you serum starve cells, they go into quiescence, but that's only one of several different changes that happen within the cell. The other is the other main one would be the production of heat shock proteins, so chaperones that facilitate protein and RNA refolding, and that could be important too.
But certainly for development, the cell cycle is very important. If you don't have G0, G1, you get very low rate of term animals.
You could say that in the nuclear transfer system,
Yes.
the empirical observation is that cells in G0
Or G1.
or G1 are more easily reprogrammed. However, whether that is directly due to the cell cycle or whether it is due to some other aspect of serum starving the cells, I think remains to be resolved.
Well, G1, the cells are not starved.
Or are they in G1?
So these cells are normally cycling, and then they do the method of the shake-off, and then the cells can come off. Those cells are not starved.
Okay, but you have like Sibeli arguing against that as well, don't you? He's got data showing that maybe it's not that important.
But the birth rates indicate on the contrary.
Okay. All right. Well, it's unresolved. I'm just telling you what I saw.
All right, any further questions? Oh, well, thank you, Steve.